We turn finally to some of the broader implications of nanoscale fluctuations in biology, particularly fluctuations in gene expression and gene splicing. This slide here shows the thumbprints of identical twins, and in case you've ever wondered, no, they are not identical. This is Cece, the first cloned cat, and her mother, from which she was cloned, and you see that they look quite different. How might such differences arise? Here is a beautiful experiment showing how simply the small number of transcription factors in a cell available to turn genes on and off can influence the phenotype of the cell quite strongly. So same genome, but different gene expression. How was this experiment carried out? Well, bugs, these were bacteria, can have circular genomes. I'm showing the two strands of DNA here in a very schematic way, which may have a promoter, a site at which a transcription factor binds. And they can be read, it turns out, both one way and the other way around the genome. Now, the rate of expression of the genes depends on the local concentration of transcription factors. Now, for genes that are used fairly rarely by the cell, the number of transcription factors may turn out to be quite small, perhaps only 10 molecules in a given cell. That means in turn, if you recall this expression we introduced in Chapter 1, fluctuations of perhaps 30% in gene expression. These fluctuations were measured by the brilliant construct of including in the gene, using recombinant molecular biology techniques, an extra piece of gene that codes for either green fluorescent protein or cyan fluorescent protein. The result then is that every time the gene expresses its desired gene product, the product is labeled with either cyan or green fluorescence. For genes required for the basic operation of the organism for which the gene expression level is high, the color is indeed an average of cyan and green. But for genes where the gene expression is low, you see large fluctuations from bug to bug. Let me turn now to an even more fascinating, in my view, consequence of fluctuations, which is stochastic splicing. You may recall in our Molecular Biology 202 slide that I said that the exons that constitute the packaged genes in eukaryotes might be put together a large number of ways, and in particular done so randomly. Perhaps the first example of a system in which this occurs are antibodies. Antibodies in the immune system are really incredible and hard to understand if one thinks about them deterministically. It turns out that antibodies are generated with an incredible array of combinations of amino acid sequences in part of their structure called the variable region. This is much like the combinatorial chemistry we talked about in Chapter 9 when I discussed the use of phage display to make variable proteins. This variability comes about from the random splicing of the exons in the spliceosome, presumably where thermal fluctuations dominate the sequence in which these cassettes are put together. The magic of the immune system is that once an antibody, a random antibody from a large library, does bind its target because the combinatorial chemistry just happens to be right, a chain of events is triggered in which that particular gene sequence is then cloned to make millions or billions of copies of the successful antibody, and that is the antibody that attacks the unknown invader in one's body. This is a picture of the structure of antibodies. It's this region here, the heads of the antibodies that have variable sequence, and are put together from these variable components of the genes for the antibodies. This is a schematic picture of the way in which the exons occur on the gene for an antibody, and it's random splicing of these various domains that results in the large number of available sequences in the variable region of the antibody structure. So simply counting these gene cassettes and asking how many combinations can be made, one arrives at something like 10 million. It turns out that post-translational modifications add an even greater degree of diversity, and so there are possibly hundreds or maybe even thousands of millions of types of chemistry available to bind an unknown invader. Here is one of my favorite examples, though, of random splicing elsewhere in an organism. So we go now from the immune system, which was perhaps the first example of this, to the development of neural networks. Neurons are long filamentous cells that propagate throughout a brain or nervous control system and form junctions that communicate, that are held together by specific sticky molecules called cell adhesion molecules. Now, it turns out that the genome for one type of cell adhesion molecule studied in the work referred to in this slide, the Downs syndrome cell adhesion molecule, for which the gene is called DSCAM, has a total of 38,016 splicing variants. The team examining this molecule wondered what would happen if they used recombinant techniques to reduce the diversity of the gene. When they did this, reducing the number of possible splicing variants to 22,176, the poor old fruit fly's neural network required for monitoring signals from the ear bristles of the fruit fly went from healthy to pathologically deformed. At first sight, this is a shocking result. You make something less random, and the outcome becomes more random. But throughout this book, we've learnt that one needs an adequate base of states for an emergent phenomenon. And if you reduce the diversity, you may not have enough complexity for the right combination to evolve. Here, the picture is that in the nascent brain, many types of connection are made by different combinations of cell adhesion molecules. Only a tiny fraction of these connections are relevant to the operation of the organism. Now, which connections they are is unknown a priori, like the immune system. But when the fruit fly, as a baby fruit fly, has to respond to external sound signals, the connections that are useful will be reinforced, and the connections that are not are atrophied. The incredible thing is that from this molecular diversity comes beautifully organized physiology. And when the molecular diversity is reduced, the correct physiology cannot emerge. So, you see that random fluctuations at the nanoscale may be a critical part of the process of putting together the conscious brains that I thank for listening so patiently to my lectures.